So, good afternoon, everyone. In continuation to the yesterday's concept related to the role of computer-aided drug design in drug discovery and drug development, we have discussed some of the basic aspects, why it is essential to use computational methods for designing and discovering the new type of drugs in the previous session. And here, yesterday we have discussed the basic aspects, followed by how homology modeling is helpful for the discovery of new drugs, and we have already utilized the software, how it can be operated in order to develop a structure of a protein which doesn't have a X-ray crystallographic structure. And another important aspect of this workshop is there is nothing but the molecular docking simulation. And this is another important area where medicinal chemists routinely use this technique in order to predict how much effective the designed molecule is or the lead molecule is. So molecular docking analysis, it is one of the most basic and important strategy that is applied in the drug discovery. And the main advantage of this is it helps us to understand how the ligand can bind to the protein through which type of molecular interactions. That is why it allows prediction of molecular interactions that will be occurring between the target protein as well as the ligand or the lead molecule or the drug molecule. And coming to different kinds of intermolecular interactions, just a second. Coming to the different type of intermolecular forces or the interactions that will be operating between the target protein as well as the ligand. They include the electrostatic interactions, that is also known as the ionic interactions. And we also, that a ligand can bind to the receptor through hydrogen bonding interactions. And whenever your drug molecule has a dipole and the dipole of the protein, both these can interact, and it is called as a dipole-dipole interaction. And we also have ion dipole interaction, wherein sometimes our drug molecule will have a ionic structure and the dipole of the receptor can. In a similar fashion, we also have dipole of a drug molecule or the ligand and ionic component in the receptor. And there are also cases where there is induction of the dipole in the target by our molecule, and it is called as induced dipole interaction. And there is also another crucial interaction known as van der Waals interactions, or it is also called as hydrophobic interaction. That is particularly seen whenever your drug molecule interacts with the hydrophobic part of the the receptor or the target protein. So coming to this electrostatic interaction, this is typically say this is your lead molecule or the ligand of the drug molecule. Whenever it has ions, because we have generally in drug molecules both basic as well as acidic functional groups. The acidic functional group, after entering into the physiological system, it will undergo the ionization process and it will convert into an anionic state. And this is the target protein. In the target protein, as it is an amino acid, as it is made up of amino acid, it has different ammonium ions or the ammonium groups, and there is an interaction between these two. This is electrostatic or ionic. This is nothing but the interaction between two oppositely charged species. Similarly, the drug molecules, majority of the drug molecules, whatever you take, they also have a basic groups. Say, for example, the drug molecules belonging to antihistaminic agents. And there are many drugs which ends with INE, you can see. All the drug molecules where there is the name ending with INE means it is a basic group, it is a basic drug, and the drug molecule containing a basic group will undergo the protonation process, and it will having a positive charge in the drug molecule. And this again, as the target is a protein which contains carboxyl functionality, it will also have the carboxylate ion. There is interaction between these two. Whenever there is an interaction between oppositely charged species, we call it as ionic interactions. In addition to that, the another most important and this is one of the vital type of interaction that we can predict by using the molecular docking studies. That is nothing but hydrogen bond. Hydrogen bonding is nothing but it's a type of dipole-dipole interaction. Here you have a dipole, here you have another dipole. And between these two dipoles, one of the dipole has a hydrogen. That particular hydrogen will be interacting with the other dipole of the target. Or your dipole of, the drug molecule can interact with the dipole of the target that contains the hydrogen. This is called as hydrogen bond. And this is one of the most promising type of drug receptor interaction that is more helpful in order to show better activity. And we have the drug molecules are either hydrogen bond donor. Hydrogen bond donor drug molecules contain hydrogen within them in the dipole, whereas hydrogen bond acceptor drug molecules have a dipole, but they doesn't have the hydrogen in the drug. That is the basic difference between a drug which bearing the hydrogen bond, which is called as hydrogen bond donor, and the other one which is called as hydrogen bond acceptor. That is the basic difference, right? And similarly, on the other side, we also have target, which may be either, sometimes it may be either hydrogen bond acceptor or it may be either a hydrogen bond donor. And this is one of the most important type of drug receptor or the intermolecular interaction. And the other type of interaction you can see is dipole-dipole interaction. That is between the two dipoles, that is the dipole of the binding site of the protein as well as the dipole of the molecule. See this dipole of the drug molecule, say typically the carbonyl compounds, and there are many other dipoles like hydroxyl group. And all these dipoles, whenever they come to the vicinity of the binding site, this dipole can interact with the dipole. Here it will be having the partial positive charge and the partial negative charge. The partial positive charge will focuses towards the partial negative charge of this dipole, and the partial positive charge will focuses towards the negative charge of the dipole. Whereas the negative charge of the dipole will be aligning towards the positive part of the dipole. This is another type of interaction which we regularly see. And there is also another type of interaction known as ion dipole. Here, your drug molecule, sometimes it may be having a dipole or it may be ion. This is also possible. Here in this case, in the example, whatever we have represented here, you see that the drug molecule contain a dipole. This is one dipole, right? Plus charge and negative charge. This is dipole, two pole, positive and negative pole. This will interact with the ionic component, particularly that is nothing but the negative charge. Whereas the negative part of the dipole will interact with the positively charged ionic component of the target protein. And in addition, we also have induced dipole interactions, where actually here you can't see any type of dipole. But whenever you have a molecule which has a dipole or charge, whenever it comes into the vicinity of this, it will induce as a dipole. If you see here, due to the influence of this particular positive charge, there is separation of the charge here. There is partial negative charge and partial positive charge. Such type of interaction is called as induced dipole interaction. That is typically seen between a charged species, and it will induce a dipole in the non-dipolar component, and there will be interaction. So overall, not a single type of interaction we can see with a molecule. We can see multitude of interactions. Not single interaction is sufficient for a molecule to fit into the receptor and show its activity. And we can get all these two or more than two types of interactions will combine, and they will interact with the active site of the protein molecule or the receptor. And depending upon the affinities or the interactions, there will be difference in the potency. One drug will be having greater potency and the other drug will be having less potency. The one which having the greater potency means it has more binding interactions and effective binding interactions with that of the receptor. If you see all these interactions are non-covalent. That means they are only having certain intermolecular forces. There is also another type of interaction known as covalent interactions, which causes the desensitization of the receptor. Will form a covalent bond with the binding site and for the formed covalent bond will cause the destruction of the receptor or change the structure as well as the function of the receptor. So that is very, very rare, and covalent inhibition is particularly seen with certain compounds like penicillins. For example, the penicillins, the beta-lactam antibiotics will causes the acylation of this serine residue that is present in the transpeptidase. Similarly, the aspirin, which is used as an antiplatelet agent, it will also cause covalent inhibition of the COX through irreversible inhibition. And similarly, you can say anti-cancer alkylating agents like carmustine, busulfan, like that, a variety of alkylating anti-cancer agents, they will also show their action by covalent inhibition. But only there are limited number of the drug molecules which show their action through covalent inhibition. But majority of the molecules, you can say, they show their action by non-covalent interactions. They show their non-covalent interaction and they will go back into the blood plasma and they will undergo metabolism and they will get eliminated. Whenever there is a covalent inhibition, it is called as irreversible inhibition, which is not useful in all the cases, and it may be responsible for the certain types of toxicities also. So, not only that, all these are the attractive forces, the intermolecular forces, the covalent or the non-covalent interactions. However, the repulsive forces are also important. The repulsive forces are important because if there are no repulsive forces operating in the nature, the entire universe will be like a single ball. We won't have a gap. We all together will get attracted and the entire universe will become just like it. That is why this particular repulsive forces are also crucial in order to show the activity also. And our software tools will help us to understand what are the different possible interactions that will be shown by a ligand or the drug molecule at the binding site or the binding pocket of the receptor. As I told, there are many type of interactions. Not all the drugs will show the interactions with all types of intermolecular forces. There are limited number of intermolecular forces that will be operating simultaneously so that the molecule can effectively. And there, before doing any experimentation, these particular in silico tools or molecular docking tools are able to predict what are the possible types of interactions that is possible with that of the protein molecule through the ligand. And if you take a drug molecule is not a single stable conformer. Say conformer in the sense, simply I can say, I am sitting now. This is one conformation. I can also stand. That is another conformation. I will be having a sleeping posture. That is another conformation. In a similar way, the drug molecule that reaches the site of action of the protein, it can have n number of the conformations. Not all the conformations are having the same energy. Say, whenever I'm sleeping, I'm much relaxed, right? And I have very good, I have very good stability. But when I am standing, I will be having less stability, right? In a similar fashion, the drug molecule will have different orientations of the different conformations. And due to these different conformations, they have different energies. Not all the one are having same energy. The molecular docking actually involves a computation process of searching for a conformation of the ligand that is able to fit both geometrically and energetically into the binding site of a protein. Say out of these 4 conformations, one of the conformations, this is high energetic conformation. If it has high energy, it can't be stable enough to bind with the receptor. Yes, sir. Say this is more stable conformer, and as it is stable, it can effectively interact with the receptor. If it is unstable, it will change its state or the geometry because of its high energy. That particular most stable. Sorry. Is it okay? Okay. And the most stable form of the molecule, we will identify using this molecular docking studies, and we can see that how it can fit into this particular receptor. So it will find the best match between the 2 molecules in the 3-dimensional space. Right. That is why we are calling it as geometrically and energetically. And this is the typical workflow for molecular docking studies. In molecular docking, we have 2 major components. One is the ligand and the other one is the target or the protein, and we need to select a target, and we need to prepare that particular target which we have selected. The software will help us to prepare the target, and we should have that target in a energy minimized conformation. Similar to the drug molecule, the protein is also having multiple conformations, right? The most stable conformation will be taken into consideration because it is the conformation where the drug molecule can bind with the protein. If protein is also unstable, the drug molecule can't, even the most stable drug molecule cannot bind. Because if it is unstable, it will orient like this, right? It will go away from this. The binding pocket may not be orienting towards the requirement of the drug molecule. If it is unstable, it is not orienting exactly according to the requirements of both the stability of the protein or the target and the drug molecule or the ligand is essential. So first we need to select the target of our requirement and also the lead molecule or the ligand, and then we will prepare both this into most stable conformation. We will transform them into the most stable conformers, and we will start docking this ligand onto the protein. And whenever we dock the ligand molecule onto the target, there occurs different types of molecular interactions, drug molecule interactions or the drug ligand interactions, and we will evaluate the docking result. In docking result, what we will evaluate is what are the types of binding forces that are operating between the target and ligand, and what is the docking score, right? And both these are the two valuable informations we can get from the molecular docking studies. The binding energy of this is important. Whenever you see a high negative binding energy, more means we'll get the values in negatives. Whenever it is in negative, the negative value is more. Say for example, you got for a compound -10 and another compound with -25. That particular compound which has -25 is more active. That means it is binding effectively with the target, right? That is called as the binding energy or the dock score molecules. And coming to the steps involved in the molecular docking studies, first we need to identify the structure of the target. As discussed in the previous session by Dr. S. Dilip Kumar, that we have a protein data, right? Where it will store a large number of the crystalline structures of the well-known protein. Scientists who have dereplicated the structures of novel proteins, they will store the crystal structures of that particular protein in the protein data bank. And we can search the protein data bank. This is the link from which we'll get the structure of the target molecule. And after identifying the target molecule, not all the space of the target molecule is required for us. There is one particular site in the target molecule known as active site or the binding site. We are much interested in that particular binding site. Next, we'll start locating the binding site. Generally, that particular crystal structure will have an co-crystal ligand, which will be acting on the binding site. That will be the typical point where we will search for, right? And after determining the binding site, we'll take our ligand and we will start docking that particular ligand into that receptor. And we can determine the binding mode or the pose of the ligand with the target. I told that here, if you see, there are different poses of the molecule, but there is only one pose that is much stable, and that particular pose can interact with the receptor effectively, right? And we will determine the binding mode or the pose of the ligand with the target. And we have different models of docking, like rigid protein docking, partial protein flexibility docking, and full protein flexibility docking. And if you see the rigid protein docking, here we will assume the conformation of the protein that cannot be changed. That means the protein's structure is rigid, and it is one of the commonly used model currently. Okay, and we also have partial protein flexibility. And I told that there are two sites, right? One is the binding site, and the other one is the rest of the molecule. That is called as allosteric site, we can sometimes say. And here we will consider the flexibility of the protein only at the binding site or the active site. That is called as, whenever we consider only this, then it is called as partial protein flexibility docking, and full protein flexibility, wherein the ligand and protein are considered to be fully flexible. So these are the different modes of docking, but typically we employ the rigid protein docking model, in order to assess the dock score as well as to understand the types of binding interactions that will be occurring between the drug as well as the target molecule. And we have a variety of molecular docking types, and we are much interested and routinely interested in this type of docking. That is, we are interested in designing the drugs and drug-like molecules of the lead compounds, and we are much interested in how the drug-like molecules of the drugs are interacting with the receptor by using this molecule. Not only that, we will also it is also applicable for studying the enzyme substrate reaction, because if you take our physiological system, there are many type of enzymatic reactions. All these enzymatic, the understanding of how the enzyme and substrate are interacting will also help us to develop novel inhibitors or activators of that particular enzyme, so that we can use that particular structure of the substrate and the nature of the interactions between the substrate and enzyme in order to design novel type of compounds that will be helpful for a variety of disorders. Similarly, we also see the protein DNA interactions or the protein RNA interactions. You know that DNA is associated with proteins. We'll call them as nucleoproteins, and the association of the nucleoproteins can also be determined by using this. How they are interacted can be determined by using this molecular diagnostic. We will also perform protein-protein. There are cases where two proteins in the physiological system will get combined, and understanding all these type of interactions, the enzyme substrate or the receptor ligand, protein DNA or protein-protein, all these are helpful in the design of new type of drug molecules for a variety of disorders. That is why the molecular docking is playing a crucial role nowadays. You can't see majority of the medicinal chemistry papers without the molecular docking, and it also has application in material sciences also, the docking studies, right? And coming to the major applications of the molecular docking, it is helpful for the determination of lowest free energy structure for the receptor ligand complex. Whenever we have a ligand complex interaction, ligand receptor interaction, that particular complex will have different energetic state, and molecular docking will help us to understand the lowest free energy structure of that particular receptor ligand complex. In addition to that, it is also used to calculate the differential binding of ligand to two different macromolecular receptors. Say, for example, a similar ligand can bind to two different type of receptors which are closely related. We can also, this particular molecular docking will also be helpful for us to understand how effectively or what is the difference in the binding between these two receptors by a similar ligand. Say, for example, if you take adrenergic agonists, the adrenergic agonist can bind at both alpha and beta receptors. Both these are close, they are having close structural resemblance. That means your drug molecule will also have similar structure. Whenever you bind at both the receptors, there is a difference in the activity. The drug molecule or the ligand will have more affinity at one particular receptor and less affinity at other particular receptor. However, it has affinity. That difference in the binding ability of that particular ligand can also be studied by employing the molecular docking studies. In order to, in addition, it is also helpful to study the geometry of a given complex. Whenever two things are happening and ligand and the target of the receptor combines, we can also study the geometry of that particular complex, right? And it is also used for the de novo design of lead generation, right? In addition, we can also design a library of compounds. Say you have taken one particular compound of common skeleton, you just change the smaller functional groups in the overall molecule. That particular small change in the functional group will help us to design a library of the compounds based on their molecular docking score. And docking is also widely used for predicting protein-protein interactions. As I said, there are many types of protein-protein interactions that are involved in different types of diseases or the disorders. Say, for example, one of the common problem is Alzheimer's disease. This is due to different types of protein-protein foldings as well as the interactions. That particular interactions can also be predicted by this molecular docking, and this will pave the way to counteract that particular protein interactions by designs novel type of inhibitors which will overcome this type of disease. Like this, not only in that, there are many other diseases like cancer and microbial infections where the protein-protein interactions plays a crucial. And we have a variety of softwares that are available in order to conduct or perform these molecular docking studies. And the first and foremost one is the GNU dock. It is a tool for docking nucleotide sequence. And we also have AutoDock, which is one of the free software, and it is commonly available. And it is a docking software commonly used for docking all kinds of protein and ligand. And this is one of the routinely used software by majority of the scientists, and particularly located in the developing and underdeveloped countries, because it is a totally free software. We can use that. And we also have other softwares like GORD, that is genetic optimization for ligand docking, and it is a genetic algorithm for docking flexible and small ligands in the protein binding sites. Not only that, we also have Flex that provides protein ligand tools, ligand docking tools in structure-based drug design. And the most important one, which we are going to elaborate today in this workshop, is Glide. It is a complete solution for ligand receptor docking. As a medicinal chemist, we are much interested in ligand receptor docking. And this is a very valuable tool in order to design and understand the interactions between the drug as well as the receptor molecule. And Glide is a complete solution for ligand receptor docking, and the Glide offers a full spectrum of speed and accuracy. That is the major advantage. And whenever you perform docking, you will be having n number of the compounds. From the n number of the compounds, by using this computational tools, you are able to identify certain best compounds. That means you should speed, and it is also accurate. That is the advantage of the Glide. There might be differences in the level of accuracy that is shown, but you can much rely on this Glide as it is much accurate, and the speed of the performance is also very high. And other advantage of this is it can be useful for high throughput virtual screening of millions of the compounds to extremely accurate binding populations. Because whenever you have millions and millions of compounds, all the compounds can be screened within less time by using this Glide, right? That is the other advantage. We have different types of compounds like semi-precision and precision, but high throughput virtual screening is the one which is not possible with all the other softwares, but Glide has that particular advantage in order to screen a large number of the millions of the compounds using this tool, right? And with this, I will transfer the session to Dilip Kumar, who will be taking you into the molecular report of some of the studies we have conducted previously, and how we can utilize this Glide software in order to dock the ligands in the receptor binding site. Thank you, and we will meet again in the question and answer session.
Good afternoon, everyone. Thank you, Dr. Basu sir, for the introduction. Now we are going to discuss some of the projects which we have done in years. Now we'll discuss some of the projects which we have done. First project is structure-based design and synthesis and activity studies of small hybrid molecules as HDAC and G98 dual inhibitors. This work we have published in Oncotarget Journal. And here we targeted two receptors. First one is HDAC and second one is G98. HDAC is histone deacetylase enzyme, and G98. These two proteins are like very responsible for treating cancer. If the suppose if the if the increasing amount of HDAC or G9A in the body means it may lead to the like overproliferation of cells that leads to the cancer. But for suppose if you are inhibiting both these enzymes, like simultaneously HDAC as well as G9A, we can treat the cancer. But there are a number of drugs for there where we can employ those drugs to treat cancer by inhibiting HDAC as well as G9A. But there are very few drugs only there. Simultaneously, they can act both HDAC and G9A. Here you can see some of the examples. HDAC inhibitor, that is SAHA, and G9A inhibitor, that is BIX01294. These are some of the clinical candidates where we can utilize these compounds for the treatment of cancer. But their mechanism of action is different. SAHA is acting on HDAC and BIX01294 is acting on G9A inhibitor. But we want a dual inhibitor. Then we thought to take what are the important pharmacophores of HDAC inhibitor, that is SAHA, and important pharmacophore of G9A. Here you can see hydrophilic zinc binding group is very, very essential for the inhibition of HDAC. There is a zinc atom in the HDAC protein, histone deacetylase protein. There is a zinc ion that binding should be there in order to inhibit that HDAC protein. Here you can see hydroxylamine group, it is in hydrophilic in nature. It can interact directly with the zinc atom so that HDAC can be easily inhibited. And there is a other core, that is lipophilic cap. Here we took lipophilic cap, that is here this group, this bicyclic ring is a lipophilic cap, and we combined this lipophilic cap as well as hydrophilic zinc with a linker. Then we got some of the hybrids, and we have screened around 20 compounds on the different cell lines, cancer cell lines, as well as we have performed enzymatic inhibition studies on HDAC and G9. Then we got some of the compounds which have shown very potential inhibition for the HDAC as well as G9. Then this is the lead compound. Here we can see compound 14 displays the desired dual activities comparable to the controls, that is SAHA and BIX. This lead compound, this compound 14 has both SAHA and BIX features. This group is phenazolene group. It is belongs to the BIX, and this hydroxylamine group, this part belongs to the SAHA. And for suppose if we, with the help of molecular hybridization, we have combined both this lipophilic as well as this hydrophilic moiety now to get the enhanced activity. Then we got very good activity, dual activity for this compound, and we want to explore how these lead molecule is interacting with protein, both HDAC as well as G9. Then we have performed molecular docking studies on both HDAC as well as G9. You can see first a picture. There is this protein belongs to HDAC. This protein belongs to HDAC. We took PDB-ID of 1P67. We took here. Then we can see the representation. This is the catalytic cavity, that is active pocket. Only hydroxylamine part only entering into the active pocket. That active pocket was filled by or occupied by only hydroxylamine part. That is, we took it from the SAHA, that is clinical candidate. But in case of other protein, that is G9A protein, the active pocket was filled by only that lipophilic cap. That active part was filled by only lipophilic cap. From this, we can say like each one molecule, it has dual inhibition. Simultaneously, it can inhibit HDAC as well as G9A. And here you can see the molecular interactions of that compound 14 with the active site residues of HDAC8. There we can see this hydroxylamine part specifically interacting with the zinc atom so that it can destabilize the protein and it can easily inhibit the HDAC protein. And also it is showing number of hydrogen bond interactions as well as hydrophobic interactions with the active site residues of the HDAC enzyme. And here you can see this lipophilic cap is occupying this active pocket, and other part of this compound 14 is showing very good interactions with the protein. And coming to the second project, that is synthesis and molecular modeling as well as evaluation of alpha glucosidase inhibition activity of 3,4-dihydroxy bipredicate. Yesterday we have seen how to create homology model. Right here you can see the template like a sequence alignment of alpha glucosidase as well as isomaltase. After this, we have developed a new homology model, and this model we have utilized it for the screening of number of synthesized compounds. Here you can see this A picture represents the homology model of alpha glucosidase enzyme, and here this is the molecular interaction diagram for the compound that is the lead compound which we are going to discuss. And here you can see this lead compound has shown very good hydrogen bond interactions as well as pi cation interaction with the arginine 3,1,2. And this work we have published in ECMC, and during that time this picture has come as a cover picture for the particular volume. And here you can see the IC50 value of the designed and synthesized molecule is 102 micromolars. And if this one is miglitol, that is already known, well-established drug, it has IC50 value of 462 micromolar. And coming to the other project, that is piperlongumine regulates epigenetic modulation and alleviates psoriasis-like skin inflammation via inhibition of hyperproliferation and inflammation. Here, piperlongumine is a naturally occurring compound, natural alkaloid. You can see there is no complex structures for both nuclear factor Kb as well as IK beta alpha. But we have done protein docking. First, we have done protein docking in between nuclear factor Kb as well as IK B alpha. After getting that complex, then we have identified what is the active site present in between the two proteins. Then we got good score for site 4, which has site score about 1.0041, as well as D score is about 1.07. And we thought that this catalytic pocket is very essential for targeting any kind of drug molecules. As well as we have we took other protein complex also, that is HDAC3 as well as IK beta alpha. And this protein complex also we have made through protein docking. But here main aim is generally this IK beta alpha will inhibits the NFKB. But NFKB is very responsible in order to regulate the inflammation. NF nuclear factor Kb is very essential for the regulation of inflammation. But IKB alpha, it will inhibit the NFKB. But our intention is that to block this kind of action. That means we are enhancing the activity of NFKB, but we are mitigating or we are reducing the activity of IKB alpha. And this structure represents the complex of both HDAC3 as well as IK beta alpha. And we can see the site 1, which has site score of 1.045. That is the catalytic pocket in between that junction. Then we have docked the piperlongumine structure in this catalytic pocket of HDAC3 as well as IK beta alpha and NF nuclear factor Kb as well as IK alpha, B alpha protein. Then we got the interesting interactions in between nuclear factor Kb as well as IKB alpha. That means in junction of this both in this complex, like both NFKB as well as IKB alpha, this piperlongumine is going there and it is sitting there and it is inhibiting the complex formation between this IKB alpha as well as NFKB. So that the action of IKB alpha and on nuclear factor Kb can be reduced. So that the reason nuclear factor Kb can easily reduce the inflammation. That studies we have performed during in collaboration with other research people. Now we are going for a small demonstration on the docking, how we can do by utilizing Schrodinger. Yeah. Here we can see the screen of Schrodinger. Yesterday we have seen like latest version, but now we are showing little older version in order to reduce the complexity. Like yesterday one, it is little slow. Here you can see this is the this is the workspace, and there are a number of options for the project, edit, view, workspace, tools, tasks, workflows, script, windows, and help. And first of all, whenever we open the software, first we need to go to master, or first go to project, and we need to change the directory. First we need to change the directory. Now I'm creating one new folder with the name of molecular docking. I choose like whatever output files will be saved on the particular molecular docking folder. Okay. In the first step, what we need to do is we need to prepare the protein. You can see after clicking this option like print wizard, we'll get the screen like this. That is protein preparation wizard. The crystal structures RSR has discussed about crystal structures. We can get the crystal structures from the PDB data bank, that is protein data bank. All the crystal structures, all the protein crystal structures will be available there. First, we need to download that PDB ID. Yesterday, we have seen how to download the PDB ID of like isomaltase of Saccharomyces cerevisiae. We can likewise we can download the PDB ID for our required protein. Then we need to browse that protein. Here you can see browse option. Then you can go for wherever you have saved that PDB ID. You can go there. And then you can import that PDB ID. You can import, like right now I don't have access for that. You can import that PDB ID. Then you will get the protein structure on this workspace. You will get the protein structure on the workspace. Then here we can see number of options, that is assign bond orders, add hydrogens, create zero order bonds to metals, create disulfide bonds, convert selenomethanones to methionines, fill in missing side chains using prime, fill in missing loops using prime, cap termini, delete waters beyond 5 Angstrom units. We'll explain what is importance of all these parameters. First one is assign bond orders. As we know, like whatever crystal structures they are deposited by scientists, we cannot completely rely or depend on that particular crystal structures. There might be some errors, like like lacking of bond order, uniformity, and some kind of small errors. We need to rectify that kind of errors. That's why first what we need to do bond orders assignment by clicking this option. Then add hydrogens. If you take the any crystal structures, there will be no hydrogen atoms. That means hydrogen atoms suppressed proteins only they will deposit. Crystal structures that only contains without hydrogen atoms only they will deposit in the protein data bank. But for the interactions purpose, we need hydrogen atoms for the protein. For the protein, we need the hydrogen atom. So we need to click this add option. Second one is add hydrogens. Then create zero order bonds to metals. For suppose if I took like any co-crystallized ligand containing protein, then if it is already interacting with the co-crystal ligand, for suppose like we have seen already one report where a zinc atom is very essential for the activity. A drug molecule is acting, binding with the zinc, atom, and it is destabilizing the protein. But suppose it is if that co-crystallized ligand is already bonded with zinc atom, is that metal order, like bond order of that particular metal will be changed. So that the reason we need to make it into zero. That is create zero order bonds to the metals. Then create disulfide bonds. For suppose some kind of sulfur containing amino acids are there which are responsible for activities, we need to click this option like create disulfide bonds. Then convert selenomethionines to methionines. It is a rare options we use. Why? Because these are selenomethionines those are not natural amino acids. And fill in missing side chains using prime. Yesterday we have discussed about prime, importance of prime. Prime is one of the module of Schrodinger where we can employ that prime for the creating homology models. But for suppose if I imported the protein structure from the PDB data bank, but there are some errors like missing of some amino acid residues or missing of some loops might be there. We cannot completely rely on the protein which they have deposited. We need to correct whatever protein they have deposited. That is the purpose. We need to fill the missing side chains as well as missing loops. Okay, entire loop can be missing. At that time, we need to use this option, filling missing side chains as well as missing loops and amino acid residues. And cap termini and delete water beyond 5 minutes. Generally, this option is we need to always make it in mind. Always we need to keep water molecules within the active site range only. Why? Because sometimes water molecules interactions also important for the hydrogen bonding, and so many drug molecules they are interacted with the protein through water. Through water they are interacting, but not sometimes they will not interact with the protein. Some drug molecules they will interact through water molecules. But we will not keep water molecules in entire protein. Why? Because it may conformational changes might occur if water molecules present entire protein. That is the reason always we'll keep the water molecules within the active site range. Okay. Then we'll click pre-process option. Then all the corrections will be done to the protein. Then second step, review and modify. Here you can see the second step, that is review and modify. There you can get how many number of chains were there in protein, or how many number of co-crystallized ligands present in protein. Otherwise, how many metal atoms were there, or cofactors, or other heteroatoms which are present in protein. Everything will be displayed in these boxes. Number of chains, heteroatoms, co-crystals, everything will be displayed. But suppose we will not target on entire protein. We require only active site containing side chains. Remaining side chains we will delete. We'll delete completely. Only we'll keep only the required chains which we are going to target. Other part of the protein we'll delete, and we'll utilize only the part which is required for the docking. This option is further review and modify. Then finally refine. In this option, we'll do first optimization followed by minimization. First we'll do optimization, then followed by minimization. That means it completely decrease the energy of that particular protein, so that we can get the lowest energy state containing protein. That is the most stable. Lowest energy state containing protein is the most stable. This is we can prepare our protein. This is the first step. Protein preparation is the first step, and then second step is now we are going to see the second step. First step is protein preparation, and second step is ligand preparation. Whatever ligands which we have designed or synthesized, we need to prepare that ligand also. Like ChemDry is there. ChemDry is one of the software where we can draw the 2D structures of the drug molecules or any ligand molecule 2D structures we can draw. From that ChemDra, we can convert the 2D structures to 3D by by saving in mole form or SDF form. Otherwise, directly we can draw the structures using 3D option, 3D molecular builder option in stored in the suit itself. Most easy thing is we can draw it in ChemDra and we can convert it into mole form or SDF file form. And here you can see, go to task, and here you can see an option that is ligand preparation. That is ligand preparation. It is the second step. I'm clicking on that. Then we'll get the window like this, ligand preparation, prep, where we need to select the ligand which we want to prepare. For suppose I want to prepare like SAHA, like one of the example for the HDAC inhibitor. I want to redock that SAHA into the active pocket of HDAC means I will draw the chemical structure of that SAHA in ChemDra or molecular 3D builder of Sodexo. I will save it in a SDF form, and I will import that SDF form in this option like file name. That means you need to import that SAHA, and here you can see do not change, neutralize, generate possible state at target pH. But suppose ionization is important. Sometimes we require ionization, sometimes no need. So most of the drugs, when it enters into the body, most of the drugs, it will get ionized and it will show its action. But sometimes they may not be innate in nature, they will not get ionized and it will show its action. So very rare drugs. Here we can see generate possible states at target pH 7 to 7 plus or minus 2. That means the average human body pH is around 7 plus or minus 2. That's why we'll maintain the same constant. Then other options like desort, generate atomers, isomers. We can generate number of isomers as well as atomers based upon the number of rotatable bonds which are present in the number of stereo centers present in the chemical structures. We can generate number of isomers as well as atomers. And sometimes we require specific isomer. Here you can see retained specific chirality. That means when we require only a specific chirality, means we need to click this retain specific chirality. That means that ligand will be in that particular isomer form only. It will be there and it will bind to the protein. And here you can see other option, determine chiralities from 3D structures and generate all combinations and generate at most 32 per ligand. This much number is not required. Mostly we'll keep like 5 and generate low energy conformations around 1 per ligand. These are different options which are present in prep module. And after completion of that prep, we need to like after clicking all the options which are present here, then we can click run option. Then the job will get completed. Then we can get the energy minimized ligand molecules. In first step, we will get energy minimized protein, and second step we'll get the energy minimized ligand molecules. Then third step is grid generation. Right. Here you can see again we need to go for task. Then here you can find docking option. Right here, there is an option like grid generation. You need to click on the then we'll get the window like this. You can see receptor grid generation. It is for active pocket identification. In previous session, Shah discussed about the importance of active site. We'll not target on entire protein. We'll be particular to the active pocket where if any drug molecule it should go to the active pocket, then only that biological response will be happen. But entire drug molecule will not act on entire protein. It will specifically act on specific binding site. For that active site identification, we'll do grid generation. Whatever, for suppose like if any co-crystallized ligand is there or any bound drug molecule is there in that protein, means we can utilize that co-crystallized ligand or bound drug for identification of active site. Right here you can see pick to identify ligand. That means whatever protein we have prepared in the first step, we need to import the protein in workspace. Then we need to click. We need to pick the molecule like co-crystallized ligand molecule which is present in that protein. And other options also there, entry option where sitemap sitemap, when we do sitemap analysis is also one of the important technique for the identification of active pockets present in protein. In that case, we'll choose entry. There is an option like entry. Then other options like you can see site. Here you can see centroid of workspace ligand. That means whatever ligand which is present in along with protein, if there is a co-crystallized ligand or drug molecule, we'll pick that ligand to identify the active pocket. Or centroid selected residues. Here we can use we can pick the some of the amino acid residues which are responsible for the activity. You can get it from the literature or any databases. There might be some note like these kind of amino acids are very responsive for the interactions. So if we target on particular amino acids, there might be a positive biological response. So in that case, we'll use that active pocket amino acid residues to to identify the active site or binding site. Otherwise, we can use supplied XYZ coordinates. XYZ coordinates also we can use. We can keep XYZ coordinates. You can get this data from the literature or any databases where they will mention the active pocket XYZ coordinates. You can enter simply XYZ coordinates, then you can you will get the box or the grid. That is the active pocket. And here you can see size of the grid also you can use. So suppose one of the molecule I have designed, that size of the molecule is bigger than that of active pocket, means simultaneously we need to increase the size of the active site also. Otherwise, it will not go and occupy the active pocket. Simultaneously, we need to increase the active grid size also. We need to increase. And constraint docking, sometimes we require constraint docking also, where we are constraint docking is mainly for we are telling ligands to go in particular conformation only and show exact interactions as previously reported. That is mainly useful for the that is the main usefulness of constraint docking. And rotatable bonds and excludable volumes, there is not much worry. And this is the third step. Then after clicking all the necessary things, you need to click run. Then the third step will get completed. You will get the zip form for glide grid. That is the third step output file. Okay. Then coming to the fourth step, that is final step. Then go to task option. There you will get docking, glide dock. You can click the glide docking option. Then you will get the video like this. Here you can see ligand docking. This is the final step in the molecular docking study, ligand docking. Here you can see first option is receptor grid base name. That means in third step, we have generated one grid, right? Third step is grid generation. First step is protein preparation. Second step is ligand preparation. Third step is grid generation. Third step output. That means we have deposited one active site containing grid. That grid we need to browse and we need to select. You can see receptor grid generation. That grid we need to browse and we need to select. And here you can see docking options like different kind of precisions, SP standard precision, HTVS like high throughput virtual screening, and XP extra precision, and standard precision and peptide docking. And for suppose if our ligand molecules are in millions, like if we are screening databases which contains more than lakhs, then we'll use high throughput virtual screening. Then job will be completed very fast. But if it is like limited, if it is in thousand likewise, then we'll use standard precision. But if it is like 100 in number, then we'll use extra precision. But most of the time, we will get the good results for extra precision mode. But the accuracy for the extra precision docking is very, very more with comparison with the other precision models. Then here you can see ligand sampling, flexible or rigid. Most of the time, our ligand will be flexible in nature. The protein might be in rigid. We can alternatively, we can change that option, ligand sampling, like it may be flexible or rigid. And these are our options for there. Then important options coming to the ligands. These settings is for selecting grid and type of docking, the type of precision we want to use. Then coming to the second one, that is ligands. Here you can see use ligands from. One option, use ligands from. That may be file or project workspace or project table. Like whatever ligands we have prepared, and we have saved in the second step, that is ligand preparation step. That files we need to choose them now. That files we need to choose. Okay. Then other option like 4, then you can click use for RMSD calculations or do not use this kind of options you can employ sometimes or avoid of clicking. Then coming to the constraints, here during grid generation, we have seen like constraints which we can utilize. That kind of, if you are using constraints, that means you are telling your ligand to make a bond with some metal atom. That means you are specifying your ligand and go and it dock along same direction. That means you are giving some suggestions to that ligand. During that cases, we are using constraint docking. We are using constraint docking, and you need to click the what are the bonds you need to require, what are the metal ion interactions you need to require. You need to click all those interactions. And this one is torsional constraints. That means you are giving exact torsional angles. Also, you are specifying your ligand. And similarity sets you can do. And final thing is output. Here you can see number of items, like number of after post docking, how many number of ligands you need to you want. That means how many how many conformations we require. That we can specify here. Sometimes we can keep 1, or sometimes we can keep 10 or 50. It varies with the ligands. And finally, we need to click run. This is the final step. That is, docking will be completed after clicking this run option. Then after clicking this run option, we will get the output files. Then we need to analyze the output files. Analyzing output files of docking is very, very essential. Very, very essential. First, we need to check with the docking score. Like if the docking score is very good, then we can give the first rank for the ligand. If for suppose whatever synthesized molecule is having a docking score of 10 and the co-crystallized ligand or any known drug docking score is 8, we can say our synthesized molecule or our new compound has more potency than that of other known drug or other known drug or co-crystallized ligand. Then we'll compare, we'll see the interactions also. We'll see the interactions like hydrogen bonding as sir discussed, like hydrogen bonding, we can see ionic bonds as well as hydrophobic bonds, water Waals interactions, pi bicating like arene interactions, and other, so many interactions we can consider, and we can say like what kind of interactions our drug molecules making with the protein molecules. We'll see all kind of all sort of things like docking score and in number of interactions of ligand molecules with the protein. Everything we'll consider, and then we can rank our ligand molecules. This is the final step. After this step, we need to analyze and we'll give the report. Like this way, we'll perform molecular docking. And with this, I'm concluding this demonstration. If you have any doubts, you can ask. And I'm requesting Basa sir to give some concluding remarks.
We are now open for questions. Please, participants, if you have any sort of questions, you can ask so that we can interact.
So, over you, President.
What is the role of
President.
What is the role of hydrogen bonds permeability? Yeah, typically, if you take a drug molecule, if it has more number of hydrogen bonds, more number of the polar groups, which contain like hydroxy groups, that molecule will have less permeability. Because in order to transfer through the membranes, the molecule should have lipophilicity, which is crucial. Whereas hydrogen bond plays a more role in, solubilizing the drug in the biological fluids, as well as it is also much helpful in order to form intermolecular interactions rather than permeability. On the other side of the coin, increasing the number of hydrogen bonding capacity of a molecule reduces its permeability. Hope the answer is clear. Any other questions? I don't think the participants have questions. Something means increase the bond, increase. Yes. Increasing the number of hydrogen bonding capacity of the drug molecule decrease the permeability. Yes, exactly.
I'm adding a small note to the SARS answer. In first project, we have seen the hybrids like which has one lipophilic group as well as hydrophilic group. That means that phenazolin pod which has a more lipophilic in nature, but that is responsible for the crossing biological membrane.
Yes.
But hydrophilic group is only responsible for the interacting with the zinc bonding. Zinc bonding, it can establish number of hydrogen bonds.
Yes.
That is the case.
Ion dipole interaction. Sir, shall we wind up?
Magma,
Hello. So good afternoon, sir. Hello. Sir, am I audible, sir?
As he failed to return the
Hello.
what is a EMI every month installment, the car was seized
Hello.
by you. Is it
Hello.
still there?
Hello.
No, I will speak on it.
No, sir, you are not audible.
249.
Hello. Sir, am I audible now?
Yes, sir. It's not clear, sir.
Okay, okay. 1 minute, sir. 1 minute.
See, the car was
There is
my
background noise.
friend's car and so I would like to purchase the car from you, but it is already seized by you.
Hello.
Yes, sir.
ఇప్పుడు ఓకేనా సార్?
Yes, yes. Now it is audible.
ఓకే సార్, ఒక 5 to 10 minutes చూద్దాం సార్. అంటే ఆల్రెడీ ఇట్స్ ఓన్లీ 3:30 కూడా అవ్వలేదు కదా సార్.
Okay, okay, no problem, sir.
And students, if you have any doubts, you can ask them, or you can drop a message in the chat box.
Yes, yes.
అంటే నేను గూగుల్ ఫామ్ కూడా ఇప్పుడే పెట్టాను సార్. నేను వాళ్ళు ఫిల్ చేయడానికి కొంచెం 5 to 10 minutes తీసుకుంటుంది కదా.
Yes, yes. Sure, sir. We are happy. We are happy to meet.
Students, do you have any questions? Please ask them. Dilip sir.
Yes, sir.
Sir, session అయిపోయింది కదా సార్ ఇప్పుడు?
Yes, sir. Completely, sir.
Okay.
Sir will some concluding remarks, like whatever we had discussed in earlier, like yesterday and today. He will.
I will summarize the things.
Okay, okay.
So overall, in this workshop, today workshop, specifically focusing on computer-aided drug design, we have discussed about the importance of computer-aided drug design. That is why it is essential to use the computers as a tool for the drug discovery process. As we have discussed, they are much helpful for saving our time as well as cost, and we will also not utilize much number of the animals, and the process is more efficient and more rational compared to that of the traditional methods of drug discovery, and that is the advantage, and that has changed the thought of scientists in many ways, where it has broad spectrum of the application, and these computational tools are helpful not only in the drug discovery process, they are also helpful in other scientific areas also, like material science and biochemistry or other fields of wherever there is molecules. Whenever you deal with molecules, computational tools are helpful. They all are decreasing the time, strength, energies, and enhancing the efficiency. That is one of the major advantage of using computational tools. And coming to the specific things we have discussed, computational tools have explored well, and they were able to develop many sort of the drugs. We have seen some of the examples like Saquinavir, Indinavir, similarly Oseltamavir, and some of the drug molecules like Norfloxacin, Losartan. All these have been developed based on two strategies of the drug computer-aided drug design, that is ligand-based drug design as well as structure-based drug design. And followed by, we have seen the outline of the computational drug design process about ligand-based and structure-based drug design. Followed by that, we have discussed about, homology modeling and how homology modeling is useful. So we said that in structure-based drug design, we need a structure of the protein. There are certain conditions where there is no crystalline structure of the molecule that is deposited into the protein data bank. In such a cases, we need to prepare our model by homology modeling technique. And already Dr. Philip has explained the softwares that are utilized for homology modeling through its how we can apply in the last session. Followed by the other important thing that is we regularly use in computer-aided drug discovery is nothing but the molecular docking studies. Today we have discussed about what is molecular docking, types of molecular docking, and different applications of molecular docking, and overall process of molecular docking, and what are the actual interactions that will be present between the ligand as well as the target molecule that are possible. And this molecular docking tools or softwares are very much helpful in order to predict these interactions prior to the synthesis of the molecules in testing. So this will cut down the cost and also the time that is or the effort which we keep in the drug discovery. That is how it is helping the medicinal chemistry. And later, you have gone through some of the projects where the molecular docking is utilized as a tool, and given positive results through our research. In addition, the other thing is the software that is Glide software. How we can use this Glide software? How we can prepare a protein? How we can generate a grid? How we can prepare the ligand? How we can optimize their conformational flexibility? All these aspects also discussed through this software. And I hope the students have learned something. And as said earlier, we are always welcome for any sort of questions or the doubts or collaborations. And we hope that it has given some knowledge, and we are trying our best to do something. And we especially thank our principal sir as well as Vishwanath sir for ligating us with the Andhra Pradesh State Skill Development Corporation. And we are also especially thankful for the AP State government for bringing such type of platform where the skills of the Andhra Pradesh State people can be enhanced, and further these skills can be helpful in their regular lives, and they can make a mark not only in our state. They can use these skills and they can apply the same throughout the country or wherever they work outside the country also. And we would like to thank you everyone, and particularly the students who have been continuously engaged in this program with a high level of patience. I am thankful to everyone. Thank you so much.
Thank you, sir. Thank you very much.
Thank you, sir. Sir, can we leave?
Sir, చెప్పండి సార్.
అది ఆఫ్ చేస్తారు, లీవ్ అవ్వచ్చు.
సార్ కొంచెం లౌడ్ గా మాట్లాడరా, లో వస్తుందా?
లీవ్ అవ్వచ్చా సార్ అంటే?
యా షూర్ సార్, యూ కెన్ లీవ్ సార్.
ఎస్ సార్, థ్యాంక్ యూ సార్, థ్యాంక్ యూ సో మచ్.
థ్యాంక్ యూ సార్, థ్యాంక్ యూ వెరీ మచ్. బాయ్ సార్.
Hello. Hello.